Hi, welcome to Biology 1. In this video, I will discuss subtopic 1.5b, describe the structure of DNA based on the Watson and Crick model. So, let me start with the discussion. In 1953, Watson and Crick has proposed the DNA double helix model. Based on Watson and Crick model, firstly, they stated that each DNA molecule consists of two helical strands of polynucleotide. This figure is representing the structure of DNA in version of zoom out and zoom in version. Notice that both structures consist of two helical lines, one with maroon color and another one is with blue color. The two different color of this helical line is used to represent two different strands of polynucleotide in DNA. Watson and Crick model also proposed that the structure of DNA contains nucleotides, in which each nucleotides in each strand are joined by phosphodiester bond. Here is the phosphodiester bond. What is phosphodiester bond? Phosphodiester bond are strong covalent bond between phosphate group that connect to the 5' carbon of one deoxyribose to the 3' carbon of the next deoxyribose of the adjacent nucleotide. Let us plot the phosphodiester bond on another figure of DNA. Third, Watson and Crick model also proposed that the DNA contain pentose sugar types deoxyribose. So take a look at this picture to spot for deoxyribose sugar. So here, the pentagonal shape with peach in color is representing the deoxyribose sugar. Since this structure is very simple, so I provide here the zoom-in structure of deoxyribose. How could we notice that the sugar or the pentose sugar is deoxyribose sugar? Look at the second carbon of the pentose sugar. If the second carbon of the pentose sugar are lacking of one oxygen atom, so the pentose sugar is known as deoxyribose. Notice that the terms deoxy means remove oxygen, means no oxygen. Fourth, the criteria of DNA based on Watson and Crick is the DNA contains 4 types of organic bases, which are adenine, guanine, cytosine, and thymine. From the figure provided here, you can notice that adenine and guanine exist as a double ring that fuse together, while thymine and cytosine exist as a single ring. Fifth, helix shapes of DNA is maintained by hydrogen bond between organic bases of opposite strand. What is hydrogen bond? Hydrogen bonds are strong intermolecular forces created when hydrogen atom bonded to an electronegative atom by intermolecular forces. That is a hydrogen bond. Let us plot the hydrogen bond on another figure of DNA. The two sugar phosphate backbone containing pentose sugar and phosphate group are located outside of the helix. Here are the sugar phosphate backbone which is located outside of the helix. 7. The nitrogenous bases or organic bases are paired inside the helix. Here are the nitrogenous bases which are located inside of the helix. 8. The nitrogenous bases of the two strands of DNA are complementary to each other. For example, adenine must pair with thymine with 2 hydrogen bonds, and guanine must pair with cytosine with 3 hydrogen bonds. So just take a look at this picture, and this picture has proven that thymine bonded or complement to adenine with 2 hydrogen bonds, and guanine are paired with cytosine with 3 hydrogen bonds. So for a DNA, since the complementary bases rules are followed, amount of organic nitrogenous base of adenine should be equivalent with thymine, and guanine should be equivalent with cytosine. Next, the last point of structure of DNA based on Watson and Crick model. The two strands of DNA must run in opposite direction, in which one strand runs 5' to 3' direction, while another strand runs from 3' to 5' direction. This concept is known as anti-parallel. At the end of this video, I hope you can describe the structure of DNA based on the Watson and Crick model. Okay, that's all from me right now. Thank you for watching.